Welcome back and happy Monday. I'll be back tomorrow, so if you're enjoying time off without me, relish the time because you'll be seeing me tomorrow. But today we're going to talk about mutations. So mutations are just any time something is changed in a DNA sequence. So if we're changing a nucleotide or multiple nucleotides or even whole parts of a chromosome, that's going to be mutations. So it's anytime there's a change in the nucleotide sequence. They're not always bad. Sometimes mutations can even be good. For example, red hair was created by a mutation, and obviously that's the best hair color you can have, so that is just a gift to humanity. Honestly, red hair is just the best hair color. So not all mutations are bad. Some of them can be really, really detrimental, but some are good, and then most of the mutations that happen are actually just neutral, so we don't really notice them. So there's two categories to mutations. There's either gene mutations, which are going to affect a single gene, so it could be a gene for hair color, eye color, skin color, height, something like that. Then you have what we call chromosomal mutations. These are going to be mutations to the entire chromosome, and those are usually ones that are more severe, just because when you start changing a bunch of DNA like that, so many nucleotides, a whole chromosome, which has like thousands of genes, it can get dangerous. So the gene mutations are usually just a single base, whereas chromosomal mutations are the entire chromosome. And then we can divide gene mutations actually up even further into point and frame shift. So point is base substitution, so they're like exchanged or something like that. So it's supposed to be an A, but now it's a G. Frame shift mutations are if we're deleting or adding a nucleotide. And those can be really bad too, because if you delete or add a nucleotide, you're changing every single codon after that. So what causes mutations? Many things can cause mutations, and we call them mutagens. So the actual process of creating mutations is called mutagenesis. It can occur randomly during replication. So for some reason or another, your DNA polymerase accidentally messes up and adds the wrong base. That can happen, and those are called spontaneous. So they just happen accidentally, nothing really caused them. On the other hand, there are things that can physically cause mutations, and those are what we're going to call mutagens and carcinogens. So they're called carcinogens because they're cancer causing, and they're called mutagens because they cause mutations in the DNA, which then leads to cancer. So this can be radiation, like UV sunlight, which is why if you go to the tanning bed, you really shouldn't ever go to the tanning bed, but it's pretty dangerous if you go because that's direct UV light. Chemicals like asbestos, tobacco smoke, all of that has carcinogens in it. Non-disjunction. So non-disjunction is when the chromosomes don't separate correctly. That can be due to environmental components, or it can just be random. But non-disjunction is pretty important, and so it's going to mess up the chromosome number. You may have one less chromosome, you may have one more chromosome, you may have two more chromosomes. It can get crazy. So non-disjunction is when your chromosomes don't separate correctly during anaphase. That's not good. That's really bad. That's going to be one of those chromosomal mutations. And then also viruses can mutate your DNA. So one example is HPV. This is the human papillomavirus, and this one can actually cause cervical cancer because it can mutate the DNA in the cervical cells, which will cause cancer. So there's so many different things out there that can be carcinogens and cause cancer. So as I said earlier, mutations can be harmful, they can be useful or beneficial, or they can have no effect. And actually most of the time they have no effect. So normally people have about 10 mutations in their DNA, and most of them just are silent, they have no effect. But mutations are really important for genetic diversity. So we talked about crossing over in meiosis during prophase 1, and yes, that's incredibly important for genetic diversity. It mixes up our DNA, all that good stuff. Mutations actually are also really important for diversity because they can create different alleles. So if we go back to that hair color example I gave where red hair was caused by a mutation, before that mutation occurred, there was maybe only 2 hair colors, brown and black. Okay? That mutation caused a new allele, meaning an allele for red hair. And so that can really provide genetic diversity to a population. So what's important to know about mutations? It's that they're not always negative. A lot of the times people think mutations are just negative. That's not true. They can be positive and they can be neutral. And most of the time they actually are neutral. So you have about 10 mutations in your DNA right now. And they're most likely neutral. So what happens if you get a mutation in a body cell versus a mutation in a sex cell? So somatic cells or gametes? Well, it does matter. There is a difference. So in a body cell, aka a somatic cell, that's not going to be passed on to your offspring. So it may cause cancer in you. So if you go lay out in the sun every single day, you go to the tanning bed every day, that could mutate the DNA in your skin cells and cause cancer. However, that's not going to be passed on to your offspring because that's due to something you did in your lifetime. So if you decided to go tanning every day and it mutated your DNA, that's not going to be passed on to your offspring because it's in your somatic cells, it's in your body cells. And if you remember, in order to make offspring, you need a sperm and an egg. So it doesn't matter about the DNA in your somatic cells when it comes to, are your kids going to have this condition as well? Are they going to get these mutations as well? However, mutations that occur in sex cells, so those gametes, those sperm and the egg, those will be passed on to your children because in order to make a child, you have to have the sperm and the egg fused together. And if there's a mutation in the sperm, then there's going to be a mutation in that child or in that embryo, that zygote. So somatic mutations can still be bad. Skin cancer, not good. However, they're just not going to be passed on to the next generation. So they're not going to be passed on to your offspring. Whereas mutations in gametes in those sex cells will be passed on to your offspring because that's what makes your offspring. So point mutations are also called substitutions. So this is when we are changing one nucleotide for another. This is just going to affect a gene. It's not affecting the entire chromosome. It's just one of those gene mutations. And it can be good, it can be bad, it can be silent. So point mutations are anytime you replace one nucleotide with another. So if you look on the example, it was supposed to be a T, but it got changed into a G. So that would be a point mutation. There's one single nucleotide that's been changed. An example of this is sickle cell anemia. So I kind of went into this a little bit last week, but sickle cell anemia is caused by a single point mutation. So one single nucleotide is changed, and when that happens, it causes sickle cell anemia. And the protein for your blood, called hemoglobin, doesn't function properly, so there's a lot of issues with transporting oxygen and things like that. So it's a pretty serious condition that's caused by a single point mutation. So one single nucleotide changed, and then you get all of these effects from it. So this is kind of looking at what it should be. So the original sequence, if you look, is supposed to be GAG. And GAG is going to code for the amino acid glutamine. With this mutation, we went from GAG to GTG. So we changed the A into a T. That's a mutation. And now it's coding for the amino acid valine. If you change the amino acid that's coded for, that's going to cause some issues. So the fact that the amino acid that codon codes for changed, that's why sickle cell causes so many problems. And if you look on the left, these are your normal blood cells. The ones on the right are what they look like if they're sickled. And so they can get stuck in your blood vessels. It makes it really hard to transport oxygen, and it's pretty dangerous. You have a high risk of stroke, things like that. So there's 3 types of point substitution mutations. What that means is if we're changing a single base, there's going to be one of 3 options. One is silent, so that, yes, we changed the base, but the amino acid actually stayed the same because multiple codons can code for the same amino acid. So in this example down here, our original DNA is TTC. If we were to mutate that into TTT, that actually, the mRNA will actually still code for lysine. So that's a silent mutation. Yes, the DNA was mutated. Yes, the nucleotide changed. However, the amino acid it coded for didn't. So that's silent. Your body will not notice it. Your body will keep on doing the same thing, keep on making the protein correctly. Next is a missense. A missense is going to cause a change in an amino acid. So here we went from TTC to TCC. That's going to change our amino acid from lysine to arginine. So that is probably going to cause an issue. And that's what's happening with sickle cell. So sickle cell is a missense. One of the amino acids changed, and then from that we get a lot of problems from that. Lastly, you have nonsense, and my thing for this is me saying stop that nonsense. What that means is it introduces a stop codon. And this one can be really bad, because if you introduce a stop codon, once that stop codon's read, the ribosome is not going to read any more of the mRNA. Even if there's way more codons that need to be translated, it doesn't matter. Once it reaches that stop codon, it's releasing the polypeptide, and it's moving on. So stop codons that are introduced prematurely can be really, really bad, because you're not going to be able to code the gene. So those usually are never good. Nonsense mutations are usually never good because they introduce a stop codon way before there should be one, and that's going to be bad. So the other type of mutations we're going to talk about is a frame shift mutation. This is if you're inserting a base or deleting a base. If you remember, we read mRNA in every 3 bases, which we call codons. If you insert or delete a base, if you insert a base, you've now shifted every codon or every amino or nucleotide after that over 1. If you delete a base, you've shifted every single nucleotide after that back 1. That's going to change what amino acids are in that gene. That's going to get dangerous because it's going to change every single amino acid, or at least most of them, and your protein is definitely not going to be made correctly. So frame shift mutations is when you're shifting the reading frame, which is those 3 groups, those groups of 3, those codons. And if you look, once this base is deleted, so this base right here got deleted, we went from GGC to GCU. And then the one after that was UCC. Now we have CCU. And so it's just going to keep happening. You're going to get the wrong amino acid, wrong amino acid, wrong amino acid, wrong amino acid after that point where something was inserted or deleted. So those can be really, really bad. So this is just the examples. Always try to include images in your notes if possible. But you have silent, which is when the amino acids don't change, but for some reason one of the bases did. So here this A got converted into a T, but we're good because that codon still codes for the same amino acid. Missense means that one of the amino acids changed. Nonsense means that a stop codon is introduced prematurely so that the gene will no longer be created after that stop codon. And then lastly, insertion or deletion shifts the whole reading frame. So after something's inserted or deleted, every single codon after that is changed. So those were all gene mutations. Now we're going to talk about chromosomal mutations, which are mutations that deal with the whole chromosome. So these are really bad because these deal with thousands of genes sometimes. And you saw what happened with the sickle cell gene. That's a single gene. It was a single mutation, and it caused really bad effects. So just imagine the effects that can happen from affecting tens of thousands or thousands of genes. There can be a lot of negative effects. So there's different types. There's duplication, which is when you're copying a whole segment of a chromosome. That may be hundreds of genes, thousands of genes. It's a big deal. So that's duplication. Inversion is when you're they're like switching. So if you look right here, this is inversion. So it's kind of like a pretzel, and then they switch the order. So it's supposed to be in the order A, B, C, D, E, F, but after they invert, it's now AEDCBF, and that's not good. Translocation is when this is not crossing over. So this is translocation. I know it looks like crossing over, but it's not, because these chromosomes should not be crossing over. They don't contain the same genes at all. So A, B, C, D, E, F does not contain the same genes as G, H, I, J, K, L. So they should not be crossing over. This could be like chromosome 1. This could be like chromosome 4. They have completely different genes. They should not be crossing over at all, but they are. That's translocation. So that usually, if this happens, the embryo is not able to develop, because this is just so dangerous and it affects so many genes that most of the time the embryo is not going to be able to develop with this one. Deletion, part of the chromosome is deleted or missing. That's, again, hundreds, thousands of genes that you just delete. That's not good. And then non-disjunction is when the chromosomes don't separate. And that's anaphase, when the chromosomes separate. So if they're not separating, we call that non-disjunction, and that means that your cells could either have an extra chromosome, so instead of 46 you have 47, or maybe you have one less, so you have 45, and that's how you get things like Down syndrome. So Down syndrome is one condition caused by non-disjunction. One of the chromosomes doesn't separate correctly.